Today, we will discuss about a very interesting and very concept-based and you can say very important chapter of biology. This will walk throughout your whole life actually. This will require for your biology portion throughout your life. That is genetics, principle of genetics. So before dive into the concept, you will have to know about some basic words about this chapter. Number one is inheritance. So what is inheritance? Passing on of characters from one generation to another generation is called inheritance. Basically, we are quite resembles to our parents. Like someone can tell us that your nose is like your father. You got your height from your mother. So this may be a height, maybe face structure, maybe skin color, maybe anything that the characters which can be passed on from your parents to you or the generation after generation. Sorry, this is called inheritance. Now you have to know that what is variation then. The characters are passed on from your parents to you, but you are not completely identical to your parents, right? You are not completely resembles to your parents. You are there is a some point that could differentiate between your parents and you. So this point of difference or this degree of difference which makes the offspring different from their parents are called variation. So what's heredity? The transmission of characters from one generation to the next generation, like from parents to offspring, is known as heredity. So now you can ask me, ma'am, inheritance means passing on of characters from one generation to another. Heredity means transmission of characters from one generation to another. So these are same. So why you are writing this in two under the two header? Because there is a basic difference. They are almost same, but not 100% same. Okay. So there is a basic difference. Basically, heredity refers to the biological process through which the traits are passed from one generation to next. This is called genetics. This is the basis of genetics. But inheritance, on the other hand, describes that particular or specific traits or the genetic material that are received from the parents. The genetic material means DNA that is received from the parents to ancestor or from the ancestors. And how are they expressed? Like which character is expressed? In which point you are resembles to your parents? In which point resembles to your father? In which point resembles to your grandmother or mother? Whatever. That particular point is now known as inheritance. So this is the basic difference between inheritance and variation. Okay. Now, Gregor Johann Mendel, who is known as father of genetics, he did hybridization experiment on pea plant. The scientific name of pea plant is Pisum sativum or Pisum sativum, anything you can say. This experiment was for 7 years, 1856 to 1863. For Mendel, whatever we come to know in our today's life about genetics, heredity, and all, there is a huge contribution in genetics is for Mendel. Mendel is a huge contributor for the thoughts, for the experiments, and the concept of genetics. For his hybridization experiment, he used 7 characters and 14 traits. Now you have a doubt, surely. Ma'am, characters, traits, character is okay because we very often use the term characters. I have this character, she has this kind of character, right? But what is trait? And what is the difference between character and traits? Basically, the inheritable features of plant and animals. Inheritable features means what is get from the parents or ancestors. So these are the features of plants or animals which are inheritable. This is known as characters. But traits means the expression of that one character. Particular one character is expressed in form of traits. Traits always occurs in pairs, and very significantly, this is a opposite pair. Okay, not similar. This is kind of an opposite pair. Okay, so this is the basic difference between characters and traits. Now, already I told you that Mendel used pea plant. There are so many flowering plants in our nature, right? But why particularly pea plant? Because he used to love mutton paneer? No. There are some reasons behind choosing pea plant, that this plant gave large sample size. It is a self-pollinating plant where you can cross 2 plants as a self-pollinator. In this plant, there is a presence of contrasting traits like tall, dwarf, axial, terminal. These are the contrasting traits we will discuss about later. And the lifespan of this plant is short. So these are the advantages why Mendel chose pea plant. Okay. So these are the 7 characters and 14 traits used by Mendel for his hybridization experiment. I am showing you through the pictorial diagram so you can understand, and the visible presentation will keep your mind. Basically, first is the character will define into contrasting pairs. What are the contrasting pairs? One is dominant and one is recessive. Okay. You can see one is dominant trait and one is recessive trait. What is dominance and recessive trait? Dominant trait means out of the 2 characters or out of the 2 traits, the one which expresses in a heterozygous. Heterozygous means not similar, right? Hetero means not similar. Heterozygous organism in F1 hybrid, which is called dominant character. And recessive means out of the 2 traits, the one which is suppressed, which is not expressed, which is suppressed, is called the recessive character. Now, the first character chosen by Mendel is seed shape. So the dominant was round and the recessive was wrinkled. Next, seed color. The dominant trait was yellow and the recessive trait was green. Flower color, dominant was purple and recessive was white. Pod shape, dominant was inflated, like smooth, and recessive was constricted. Pod color, yellow was the dominant character and green was the recessive character. Flower position, axile is the dominant character and terminal was the recessive character. Stem height, tall, for dominant and dwarf for recessive character. Now, the very first experiment was done by Mendel is monohybrid cross. What is monohybrid cross? Mono means one, right? Mono indicates one. So the cross or fusion involving plants differing in inheritance of one contrasting feature. Basically, a cross involving one contrasting feature is known as monohybrid cross. Okay, here is a diagrammatic representation of monohybrid cross, but I will practically show you. Just a minute. So Mendel chose for P generation. P generation means parental generation. One plant which is capital T, that means purely tall. And the other plant was small t, which is purely dwarf. So we can indicate that this is tall and this means dwarf. So in parental generation, Mendel chose two characters. One is parental characters for tall, purely tall, and one is purely dwarf. Now, you will receive the gametes, right? You will receive the gametes from this tall plant. You will receive all gametes that are tall in nature. And from this purely dwarf plant, you will receive all gametes having purely dwarf. This will be crossed and the fusion of two gametes, will give tall plant. Capital T, small t indicates tall because here dominant character is capital T, right? Which is dominant, we will say that character will be expressed. So this plant obviously is tall. And this will be self-pollinating by a tall plant. Okay. Now, this is the F1 generation. F1 generation means the full form of F1 generation is filial 1 generation. So what is first filial or filial 1 generation? The progeny or offsprings produced from a cross of two parents. Like you can see here are the tall parent and a dwarf parent. You will get the gametes tall and dwarf, and when the fusion of two gametes gives us this tall plant. So this is F1 generation, right? Now there it will be a F2 generation after the cross of F1 generation. So F2 generation means the progeny resulting from this self-pollination. This is self-pollination or inbreeding of F1 individuals. Like tall plants is fused with another tall plant is called, we will get the result. The results will be called F2 generation. Okay. We will see what will happen in F2 generation. I am using a separate page because I have to give a space for F2 generation, because in F2 generation we will make a square-like structure which will known as Punnett square, okay, which is known as Punnett square. Now, first, here you see the gametes will be represented here, okay? The gametes will be represented here, and capital T from one gamete, small t is from one gamete, capital T, small t. Okay, now fusion takes place. Capital T makes tall plant. Capital T, small t makes tall plant but recessive condition, heterozygous condition. Capital T, small t obviously makes tall plant, but this is also a heterozygous condition. Small t makes dwarf plant. Now we have to characterize them. Okay. Capital T, this is tall. Capital T, small t, this is tall but heterozygous condition, not purely tall. Capital T, small t, this is also tall but heterozygous. And small t, this is purely dwarf. So there is two types of ratios. Phenotypic ratio. The phenotypic ratio will be 3:1. Why? You can see there are 3 tall plants and 1 dwarf plant. Phenotypic means the outside conditions of a plant of any animal. So the phenotype means the outside condition. Like we are visibly indicate that 3 tall plants are there and 1 dwarf plant. So the ratio will be 3:1. And another ratio is genotypic ratio. Genotypic means the characteristics of gene included, right? So genotypic ratio will be 1:2:1. Why? 1 purely tall, 2 for heterozygous tall, right? This is not purely tall, capital T, small t. There are 2 small, capital T, small t. They are heterozygous in conditions. And 1 is for purely tall. So genotypic ratio will be 1:2:1 and phenotypic ratio will be 3:1. So this is the cross Mendel proposed, which is known as monohybrid cross. So after monohybrid cross, we will get the first law of inheritance, and it is also known as first law of dominance. Actually, law of dominance, first law of inheritance and law of dominance, where we can see that characters are controlled by a unit, a discrete unit, and Mendel named this unit as factors. And factors occur in pair. We already wrote capital T, not one capital T, because this is the expression of allele, the 2 contrasting genes. Okay, so it is occurs in pairs, and in heterozygous condition, one of them will dominate. We saw already that capital T will dominate over small t. When capital T, small t present, it will be represented as a tall plant. Okay, so basically law of dominance states that in a dissimilar pair of factors, one member of the pair dominates the other one. Basically, which will dominate is known as dominant character, and which is suppressed is known as recessive character. This is the first law of inheritance or law of dominance. From the monohybrid cross, we will see another law, which is the law of The second law of inheritance or law of segregation. Here we can see the law of purity of gametes, and there is no blending between gametes. What will this law state? That at the formation of gametes, the two chromosomes of each pair separate or segregate in two different cells, which form the gametes. Basically, when the gametes are formed, the two chromosomes from each pair not occur in one cell. This will segregate. This will separate into two different cells, right? And this will then fuse to form gametes. This is second law of inheritance, which is known as law of segregation. Basically, these are the two laws which is proposed by Mendel from his monohybrid cross on Pisum sativum or pea plant. Okay. So this is it for today. I hope you understand well. Thank you. Thank you so much.